All right. So we're talking about the nucleus. Let's start looking at the nucleus and see what is special about it. So here is the structure of a nucleus. Okay. We have parts like the nuclear envelope. Let's see if we can point it. Okay. Okay, we have the nuclear envelope here. What do you think is the function of the nuclear envelope? What do you think is the function of a nuclear envelope? Who knows? Who would love to try? Becky, would you love to try on giving us the function of a nuclear envelope? Like the word envelope, what does it do? The word itself, envelope.
An envelope protects the inner equipment.
Correct, right? So even on the nucleus, the nuclear envelope is for protection, right? Protecting the inner parts of the nucleus. And also it has pores, like the nuclear pores, right here. And these ones allow the entry of material into the nucleus, entry and exit of material. For example, we have materials that enter and exit the nucleus, like RNA, okay, RNAs. For example, the vaccine that we're taking right now is made up of mRNA, but that mRNA cannot enter the nuclear envelope because this entry and exit requires an enzyme, right? So scientists say it cannot enter the nuclear envelope. So it can't get into our nucleus and interfere with our DNA. It remains in the cytoplasm and it does whatever it wants over there. It doesn't interfere with our DNA. But there are some materials, other RNAs, that have specific enzymes that facilitate them to enter the nuclear envelope through the nuclear pore. Okay, that's just an example for you. We have other parts like the perinuclear space. Okay, that is the space between the nuclear envelope, and we have the heterochromaty. Okay, that's the material around the nuclear envelope inside the nucleus. Okay.
Cells are the smallest living units of an organism.
There's a video playing. Let me first mute
All
it.
cells have three things in common, no matter what.
Let me first mute this video that is playing. I want to mute this video because we're not going to watch it now. We shall watch it later. Okay, let me pause it right there so it can even load. I open them so they can load so that we don't have to wait for internet when we start watching them. Okay, hopefully this one doesn't play anytime soon. All right, here it is. Let me pause it. Okay, let me wait for that to play. All right, we were saying, we're talking about the parts of the nucleus, and we're on the heterochromatin, and we say that is the material that surrounds the nuclear envelope inside the nucleus, and then the perinuclear chromatin. The perinuclear chromatin is the material that surrounds the nucleolus. Okay, this material right here, we call it the perinuclear chromatin. Now, all this chromatin is made up of a certain molecule. Who can tell us that molecule that makes up this chromatin, whether it is heterochromatin or perinuclear chromatin? What material, what molecule is that? We also have the intranuclear chromatin. What molecule is that? Who knows? Who can guess? What molecule is found inside the nucleus? It's a very prominent molecule. Very prominent. Kwagala, what do you think that molecule is? Yes?
I think it's DNA.
Yes, it is DNA, Becky. Well done. So DNA makes up all this. So inside the chromatin, the material that surrounds the DNA, we call it chromatin, all right? So inside that chromatin, there is DNA. So whatever chromatin you're seeing here, be it enterochromatin, perinuclear chromatin, intranuclear chromatin, all that is just containing. We also have euchromatin here. All that is containing DNA. All right. Remember, DNA is wound, or I don't know the other word to mean wound. Okwezingazingira muno. I don't know if you all understand Luganda. It is wound around here, okay, inside the nucleus to form that chromatin we're talking about, okay. And then we have around the nuclear envelope, we have the endoplasmic reticulum. This surrounds the nucleus. And on it, since it is rough, we have the ribosomes. Shall look at the functions of, I think we have looked at the functions of ribosomes. Who can tell us the function of the ribosomes? Who remembers? Also have the nucleolus in here, is the inner part of the nucleus. And then the inner membrane and outer membrane. What is the function of the ribosomes? Who knows? Function of ribosomes. Ribosomes. Who remembers? Xavier, do you remember? Or Becky? Function of ribosomes. You need to go back and revise that unit that I told you about, the unit before this one, because it is where everything about that is.
Master, I think the ribosomes, they are the site of protein synthesis.
That is 100% correct, Becky. Well done. It is the site of protein synthesis. So if you have forgotten the functions of most of the parts of the nucleus, please go back to that unit and revise. So that is correct, Becky. It's a site of protein synthesis. Good job. Now, let us look at the description of the nuclear structure. It is enclosed and bound by a double-layered membrane, okay, which we call the nuclear envelope. The outer membrane is connected to the endoplasmic reticulum. A fluid-filled space, the perinuclear space, exists between two layers of the nuclear membrane. We've seen that. And the nuclear membrane is perforated by nuclear pores. We have said materials like RNA pass through these nuclear pores. Is enclosed within the inner membrane of the nucleoplasm and nucleolus and chromosomes or chromatin. We have talked about chromatin as where we find our DNA. Okay, the other word for them is the chromosomes. The nucleolus is dense, spherical shaped structure. Okay, that's the nucleolus inside the nucleus. And then we have the chromosomes, which is the chromatin. They are thread-like structures, okay, in the nucleus. We have the heterochromatin, a Steindacky, genetically inactive, and it is tightly packed in the nucleus. Euchromatin loosely packed, genetically active, and enriched. So the active part of DNA, the inactive part of DNA or the inactive part of the chromosomes, we call it the heterochromatin. And then the euchromatin is the active part. So if you ever ask the difference between heterochromatin and euchromatin, the euchromatin is the active part. Okay, it's genetically active, meaning it codes for certain proteins. And then the heterochromatin is inactive. It codes for none of the proteins that we need in our bodies to perform various functions. So that's the difference. Any questions about the description of the nuclear structure? Is there something you've not understood? We can even go back to the structure if there is something you have not understood here. Any questions?
Yes, teacher, I have a question.
Yes, please.
Like in paper 2, if I'm asked to describe the structure of a nucleus, do I state, do I give my answers the way I've given it, or I have to add more flesh on what I'm putting?
You don't have to add any flesh because that will be just English. In biology, we look at the keywords. So just include all these keywords in a paragraph or two, okay?
Okay.
When you look at how we mark paper 2, we mark keywords. Do not put a lot of English in your explanations to be useless, right? Just put the keywords in the paragraph as you describe, right? So just remember only the keywords in here. Like if you're describing the nuclear envelope, make sure that you're saying it is double-layered or double-membraned, right? If you're talking about the endoplasmic reticulum, be specific and say it is rough endoplasmic reticulum and things like that. So you just have to be specific with your keywords, and that's all that matters in biology, especially paper 2 where you're asked to describe things. Is that okay?
It's okay, sir.
All right, that's a good question. Any other question? Any other questions? Okay, if there's no any other questions, we can continue. Let's look at the functions of the nucleus. So the first function of the nucleus is that it controls hereditary features. okay, of an organism. It controls protein synthesis, cell division, growth, and many others. It stores DNA, which is the hereditary material, stores proteins and RNA. It's a site for transcription in which messenger RNA are produced for protein synthesis. Nucleolus produces ribosomes, which are the protein factories. Those are the functions of the nucleus. If you find any, you can always add on. Okay. Any questions on these functions? All right, let's look at the adaptations of the nucleus. DNA is long in order to store many genes. Okay, this is why it is just wound into the nucleus in form of chromosomes or chromatin. The nuclear membrane has pores for exchange of DNA and RNA between the nucleus and the cytoplasm. We talked about this. It's not DNA goes through. The DNA cannot move out of the nucleus, but the RNA can move out. In the types of RNA, we have, for example, ribosomal RNA, we have messenger RNA, which other one? tRNA, okay, transport RNA. So the RNAs can move around, all right, because for them, the purpose is actually transcription and things like translation. So they need to move into the nucleus or out of the nucleus into the cytoplasm, depending on what they're working on. So that's the function of the nuclear pores and how they adapted to their function. Presence of the nucleolus enables production of ribosomes, which are protein factories. Becky said this about the ribosomes, that they're protein, sites for protein production. So the nucleus is adapted in such a way that it contains the nucleolus to produce ribosomes, which in turn produce proteins. It's also adapted in such a way that the nuclear envelope isolates the nucleus from interference by processes in the cytoplasm. So the nuclear envelope is able to protect the nucleus or the materials in the nucleus from interruptions from the reactions that take place in the cytoplasm. Also, the nuclear pores are very, very narrow to regulate the entry and exit of substances. Like we said, even the entry and exit of substances, sometimes, in fact, most times, it is facilitated by enzymes. Without enzymes, there will be no entry or exit into and out of the nucleus. So that is very, very important. Now, let's see if our video has loaded. It couldn't load here, so I opened it in a different browser. Hopefully it has loaded. Okay, let's check it out. It's loading, but very, very, very slow.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. there are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, yeah, well done. That is a video that tried to explain to us in details what the nucleus is made up of and what some of its functions are, but also it went into detail in explaining other organelles. Now, as far as the nucleus is concerned, can you tell me what you picked out from this video? What is something that stood out for you in this video? What stood out for you, especially about the nucleus? The video mentioned a couple of things about the nucleus. Who wants to go first? Xavier, do you want to go first? What stood out for you? Can you guys hear me?
Yes, we can.
Yeah, I wasn't sure because you're very, very silent. Now that you can hear me, Becky, what stood out for you mostly?
Sayo,
Okay,
internet
what?
breaking.
My internet is breaking.
Yeah.
Sorry. I was asking what stood out for you in the video about the nucleus.
From nuclear, I pay.
Maybe your internet is breaking also. I don't know if it is yours or mine. Becky, can you hear Xavier well?
I can, but his network is breaking.
Genetic material.
Yeah. Xavier, we can't hear you well. Becky, you can go ahead. It's even off. Becky, tell me what stood out.
What stood out for me was this part when they were like the outer membrane is continuous with the endoplasmic reticulum, and they were like, its surface often has ribosomes. So and this makes it okay to appear rough. And also I think I will give that.
Okay, well done. Yeah, that's something very nice that stood out for you. It's very, very good about the plasmic reticulum. Okay, so if you had gone off, your internet was poor. You can tell us now what you thought of the video. Hopefully we can hear you now. Yes, Xavier. Okay, if you can't hear us, that's fine. The other organ I wanted us to talk about today is the mitochondria, or the mitochondrion. What is the function of the mitochondria? Becky, what is the function of the mitochondria?
The mitochondria is a site. It's a site for pyruvic acid oxidation by the Krebs cycle. Okay,
very good. It is where the aerobic respiration takes place, and the part of aerobic respiration that takes place there is the Krebs cycle, where the pyruvate is turned into the ATP. So you're very, very correct. Very correct. Good job. So here is the longitudinal section and transverse section of the mitochondria. The mitochondria has one of these special structures is a circular DNA. It has ribosomes. It has this tact pedicle and the matrix, the crista, which are the foldings, okay? And then the inner membrane and the outer membrane. Those are the parts of the mitochondria, okay? Mainly, its structure has a diameter of about 0.5 micrometers to 1 micrometer. Also, it has double membranes. It has a matrix, which is fluid filled with several enzymes and ribosomes plus circular DNA. Each member of a phospholipid bilayer with each membrane has a phospholipid bilayer which is variable phospholipid composition on protein to lipid ratios. We can look at the adaptations. A double membrane isolate the mitochondria from the interference of the cytoplasm reactions. Small size gives large surface area to volume ratio for rapid uptake and release of materials. It has the matrix that contains enzymes for the Krebs cycle. Inner membrane forms side cristae to increase the surface area for electron transport. Cristae, we have said the foldings, the inner foldings, the finger-like projections, yeah? The narrow membrane space enables hydrogen ion concentration gradient to rapidly establish for chemosmosis. So when the gradient is created, then materials can move in and out of the mitochondria. The inner membrane contains molecules of electron transport pathway. Remember, after the Krebs cycle, we go to the electron transport chain where we see energy being produced as the electron moves from across the gradient. DNA is present to act as genetic material for synthesis of some protein and many ribosomes for protein synthesis. So those are some of the adaptations. I want you to continue with this unit. Go through all these. There are certain videos that I've put for you to watch. Make sure you watch them. And then when you're done going through, you can do the activity. So when I come back the next time, I'll make sure we go through everything else, but please, meanwhile, you also try going through. I have an attachment here, which is just a couple questions you can answer and submit whenever you're done going through the unit. Is that okay?
Yes, Master.
For the remaining time, if the internet allows, yes, if the internet allows, we can watch this video about mitochondria. So let me see if it can load. If it can't, you can always watch it in the unit. Luckily, it is here. Let's see if we can watch it.
Mitochondria are organelles found in eukaryotic cells that play an important role in the production of ATP, the universal energy currency used in all cells. Most mitochondrial proteins are transported from the cytosol into mitochondria through specialized protein translocator complexes. Interactions between these complexes bring the outer and inner mitochondrial membranes into close proximity. The successful targeting of many proteins that reside in the intermembrane space requires translocator complexes that cross both the outer and inner membranes. After entering the inner membrane translocator, many of these proteins do not completely cross the membrane, but instead are released into the membrane and diffuse laterally. They then remain embedded in the inner membrane or are cleaved, releasing a portion into the intermembrane space. Very few resident proteins are simply transported across the outer membrane translocator complex. Matrix proteins do not transit through the intermembrane space, but are directly transported from the cytoplasm across the interacting outer and inner membrane translocators. The protein-rich matrix contains enzymes necessary for cellular respiration, a process during which carbon fuel molecules are oxidized and reduced electron carriers are produced. Invaginations of the inner membrane, called cristae, contain 4 large protein complexes that harvest electrons from these carriers. Complex 2 accepts pairs of electrons from succinate and transfers them through a series of redox centers to coenzyme Q. The lipid-soluble coenzyme Q is reduced by electrons harvested from complexes 1 and 2, and then diffuses through the inner membrane, transferring its electrons to complex 3. In complex 3, electrons are transferred through 2 distinct series of redox centers, which allow them to cross the inner membrane one at a time. Finally, electrons are accepted by cytochrome C, which carries them to complex 4, where they are transferred through another series of redox centers to their final acceptor, a molecule of oxygen, that combines with hydrogen ions to form water. Electron transport in complexes 2, 3, and 4 is coupled with the pumping of protons from the matrix to the intermembrane space. The resulting electrochemical gradient across the inner membrane is called the proton motive force. Protons flow back into the matrix through a component of the ATP synthase called F0. The membrane-embedded ring structure of F0 binds protons in the intermembrane space and releases them on the other side of the inner membrane into the matrix. Proton flow drives the rotation of the ring structure, which in turn leads to the rotation of the F0 central shaft. The rotating shaft sequentially contacts the 3 catalytic subunits of the ATP synthase F1 complex, altering the subunit's affinity for ATP and ADP, and catalyzing the synthesis and subsequent release of ATP. Most of the ATP synthesized in the mitochondrial matrix is consumed outside of the mitochondria. but the inner membrane is impermeable to ATP and ADP. Therefore, an ATP-ADP carrier is responsible for the coordinated import of ADP and the export of ATP across the inner membrane. In cells, mitochondria are distributed near sites where ATP and other mitochondrial metabolites are in high demand. Mitochondria move along cytoskeletal structures such as microtubules and can undergo dynamic changes in shape, including both fusion with other mitochondria and division. Perturbations in the dynamic behavior of mitochondria and alterations in mitochondrial membrane permeability are associated with the early stages of programmed cell death.
Okay, so in this video, what I wanted you to pick out is what happens in the different parts of the mitochondria. You don't need to know the names of the subunits, the F1, the F0, the rotating shaft. You don't need to know that. You'll only get to know that when you study a lot of biology at the university. But for now, just have to know what happens in the matrix, okay? What happens in the cristae, okay? About the electron transport chain and all that. And that's it. Just the formation of ATP, and that is what is required for you to know about the mitochondria. Thank you for attending today. Make sure you go through the rest of the unit and also attempt the activity and submit your answers. I would love to know what your answers are. Well done. Any questions before we go? Any questions?
No?
All right, well done. I'll see you soon. Bye-bye.